The image you're looking at may appear to be a film of a living human heart, but it isn't. It's a highly sophisticated computer program using tens of millions of separate equations, combining graphics and mathematics to create one of the first of a new generation of simulations called virtual organs. These are set to become some of the most powerful tools we have in the fight against disease, with scores of uses from disease diagnosis and drug design to training and research.
My involvement in modelling the heart and other organs began over 46 years ago when I was working in London. I was the first person to build a computer model of the activity of individual heart cells as they were beating. And that was based on experimental information that we obtained on the nature of the processes occurring in those cardiac cells. Now, those models were very simple and they ran on very simple computers. Nowadays, 40-odd years later, we have masses of more information, thousands of laboratories all over the world accumulating experimental information. The models have become very much more sophisticated, and most importantly, they've been incorporated into models of the whole organ so that you can actually see the organ itself beating away.
The virtual heart allows doctors to study the effects of a heart attack in controlled circumstances without endangering a real patient. And unlike real life, in which a heart attack can take just a few seconds, the progress of the attack in the virtual heart can be slowed down, speeded up, and repeated at will, enabling scientists to measure exactly what is happening.
They've become sufficiently sophisticated that they are now used in pharmaceutical research and for assessment of drug action by regulatory agencies. 10 years ago, or nearly 10 years ago, I was involved in using one of these models to help a major pharmaceutical company, Roche, to process the drug application that they were making to the Food and Drug Administration in Washington at that time, and we successfully helped them to get their application through. And there have been other examples more recently where companies like Novartis have used the model to clarify the actions of the drugs that they are developing. So the work is already partially achieving the object of enabling some of the work that is normally done on animals to be done instead on computer simulations. But I should emphasize this is early days. We can't yet look to a time when you could replace animal experimentation entirely by computer simulation. But clearly the long-term goal would be to achieve a situation where, just as in the aircraft industry, you don't build an aircraft before simulating it on a computer, in the future people will not build new compounds, new drugs, new medical devices without simulating their actions on the various organs of the body.
Virtual organs such as this one allow researchers to develop and test new drugs faster, more confidently, and at less cost.
One of the best ways of finding out how a new drug will behave in the human body is to give it to volunteers at a minute, safe dose. Then, by using incredibly sensitive measuring devices, scientists can monitor where the drug goes and how it is broken down in the human system. Microdosing is an exciting new breakthrough in drug testing. It uses machines so sensitive that they could detect a litre of liquid after it has been diluted in all the oceans of the world. Acceleron is the world's leading microdosing company, of which Professor Colin Garner is the founder and chief executive officer.
Now, why is microdosing of interest? Well, it's of interest because it allows us to get human metabolism information much earlier, much more safely, and with minimal use of animals. Now, the great attraction of microdosing from a drug development point of view is that you can take your drugs into humans with much reduced costs and much reduced time compared to classical, traditional so-called phase 1 methods. So typically, to take a molecule into man using classical phase 1 methods might cost as much as £3 to £5 million and take anywhere between 12 and 18 months. Whereas using the microdosing approach, we can do the same type of study at much less cost, typically around about £250,000, and get the results in 4 to 6 months. So there is a time and cost saving to the approach, and it's been estimated that by 2008, as many as 40% of pharmaceutical and biotechnology companies around the world will be using microdosing. 2010, nearly 90% of all companies have stated that they will be using microdosing routinely in their drug development program.
These new technologies enable drugs to be developed and tested specifically for humans. Yet there is still a vocal strand of opinion which insists that new treatments will only come from research involving animals, and that it would be unethical to expose people to drugs without first showing that they are safe in animals. But this view is increasingly questioned by members of the medical profession. Kathy Archibald is the director of Safer Medicines Trust, who commissioned a survey of family doctors.
We commissioned one of the world's largest market research companies to conduct a survey of 500 GPs across the UK. The survey found that 83% of GPs share our desire to see the medical value of animal tests evaluated scientifically. That's an extraordinary level of concern. I know from my own experience as a scientist working in drug development and as a patient with a serious illness that much animal research is simply irrelevant to human medicine. What happens in animals doesn't matter. It's what happens in people that counts. My disease was in the pancreas, which scientists have just discovered is so dramatically different between rodents and humans that studying mice and rats is futile. The object of study has to be human. We acknowledge that there's no such thing as a drug that will be absolutely safe for everybody, but we do believe that drugs could and should be safer than Vioxx, for example. And we believe that one reason why clinical trials are risky is that very often, instead of information about safety in humans, all we have is an assurance of safety in animals, which, as the story of thalidomide should have taught us, means very little. The fact that 9 out of 10 drugs fail in clinical trials proves that point very clearly. So does the fact that prescription medicines are our 4th biggest killer after cancer, heart disease and stroke. We believe that drug trial volunteers should be protected from risk by making sure before clinical trials that the new drug is as safe as possible, something that may well best be established by a battery of human-based tests such as those we've just heard about.
We've heard from the experts about the potential of the latest methods for assessing new drugs. But are the people responsible for legislating for drug safety listening? Many politicians are concerned about animal experimentation, but believe there is a dilemma between protecting animal welfare and advancing medicine. Yet there is growing evidence that favouring the state-of-the-art methods we've seen in this film would benefit medical progress as well as laboratory animals. Tony Benn was a Member of Parliament for over 50 years and a Cabinet Minister for 15 of those. He's given the matter some serious consideration.
A lot of doctors and scientists are now challenging the scientific value of the use of animal testing for human purposes. And these arguments have to be taken seriously. 250 members of Parliament have signed a motion in the House of Commons calling for a government inquiry. And that call has the support of a lot of people from the medical profession and, of course, from the public as well. And what is really required is a proper scientific evaluation of the methods that are used to decide, or make it possible for us to decide, what is the value of animal testing. And it's never been done before, and there's nothing whatever to hide, so I can't see any case against it. This is not an argument against science, quite the opposite. It's an argument for bringing modern scientific thinking to bear on an issue that has in the past been simply described as if it was those who in favour of animal rights against scientists. It is actually a very important scientific development. And all that we want to see is the government to set up a review where these arguments can be properly evaluated in a scientific way, because quite apart from anything else, thousands of people suffer from the side effects of drugs, and some of those ill effects might be averted by using better methods. And the government should now, in my opinion, make this a priority, because if they did that, then this argument could be settled in the best possible way for the benefit of everybody concerned.
Safer Medicines Trust believes the government should commission an independent scientific comparison of the results from animal tests with those from a battery of human-based tests to see which more accurately predict the human response. Most MPs and GPs support this idea. The outcome couldn't be more important for those of us who await new drugs for ourselves or our loved ones, or for the volunteers who take part in clinical trials for the benefit of society and medical progress. We hope that this short film has offered a glimpse of the many new techniques that are paving the way to safer medicines for us all, thanks to their focus on human biology.